Hello, Sonalika. Could you please confirm if my voice is audible?
Hi, Shrigori. Yes, we can see your screen and your voice is also.
All right. Thank you so much. Hello, everyone.
Hello, everyone. We'll be starting in 5 more minutes. Thank you for joining. Maybe in the chat section, you can please introduce yourself and tell us about your research interests and your background. Thank you.
Hello, everyone. A very warm greeting to one and all. I would like to welcome you all for today's webinar on Omics Logic Precision Medicine Summer Training Program. My name is Sri Krishnakumar, and I'm a research intern at Biomedical. I'll be your host for today's webinar. So before proceeding further, let us discuss what this program is all about. The Precision Medicine program is designed to introduce students and researchers to the field of bioinformatics in precision medicine. In this program, we will explore how the various omics data types generated in these studies can be analyzed and integrated to study the basic biology associated with viruses and their host, cancer onset, development, and outcomes. This online program also includes weekly sessions with an instructor, online tracking, and a review of student progress, access to all the resources and materials online for self-paced learning, access to expert guidance and problem solving, educational and research pipelines, domain data analysis on our big data server for 15 days. In this webinar, we will discuss about the overview of the precision medicine program, the current applications of bioinformatics in precision medicine. We will also take a look at the detailed curriculum of the program. And finally, we will discuss about the registration and certification process. So before going further, what is precision medicine? The precision medicine is an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. This approach will allow doctors and researchers to predict more accurately which treatment and prevention strategies for a particular disease will work in which groups of people. It is in contrast to the one-size-fits-all approach in which disease treatment and prevention strategies are developed for the average person with less consideration for the differences between individuals. After decades of research, we are poised to enter a new era of medical practice where detailed genetic and other molecular information about a patient's cancer is routinely used to deploy effective patient-specific remedies to treat it. As we are about to enter the era of precision medicine, the biology is now seen as a field of data science. Biology is rapidly acquiring the character of data science. Billions of data on genes, proteins, and other molecules are compiled in large files and systematically studied. Analyzing this data will lead to more knowledge and understanding about living organisms, including human health, biotechnology, crops, and livestock. This program will give an overview of bioinformatics for students, biologists, and clinicians. The influx of data, associated methodologies, and data-driven research makes bioinformatics for life sciences as prominent as the emergence of computers for the tech industry. The goal of omics logic programs are to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. With so much data available on any biological topic in question, running an independent in silico research project over the summer seems like a great opportunity. But let me ask, do you have the resources to complete your big data analysis? Do you have a mentor that can help match the right analysis method to the research question? Do you have a community to keep you accountable and provide you with feedback? If your answer to these questions is a no, but you are motivated to learn, we are offering a special research fellowship program where you will be matched with a research mentor, get access to training resources, and big data analysis tools to get you there. After completing this program, you will know how to do the following. That is, read and understand bioinformatics literature, perform basic NGS data processing steps, analyze data and form hypotheses, prepare data for machine learning analysis, cross-check signals with gene databases, know what type of data is required to answer scientific questions, create your own bioinformatic analysis, present your results in an organized way. All these skills are essential in bioinformatics and some can be translated to other fields as well. The Omics Logic portal gives students options for analyzing a given dataset, code-dependent paths or code-independent paths. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor guide programs, where experts share their experience and support research projects developed by you. After using the Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge and apply that in a project. Then check their comprehension with levels and badges, certificates of completion, and tests. Similarly, faculty should be able to assign the Omics Logic portal to their students with the understanding that it will give them holistic and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and on to designing their own question-based project. Now, let us start by signing up on the learn.omicslogic portal. This portal, education portal, will help you to get started on your bioinformatics journey. So I'll pause for a couple of seconds so that everyone can sign up on the portal. Ojasvi has shared the link. So once you are done signing up on the portal, please let me know and I'll proceed further. All right. So once you have signed up on the portal, this is how the completed profile would look like. In the completed profile, you will have an updated profile picture. You should also link your social media handles, and that will include either Facebook, LinkedIn, Instagram, or ID, or Twitter. You can also mention your educational background and research interests so that a community can reach out to you. The mentors are also able to track your progress on the activities section and help you with any technical queries that you have. After you register, you will be redirected to the courses tab. Here you can search for any course title, use the tags to find series of resources around a topic, or choose one of the collections down below. In this session, in this video, we will look at the Getting Started collection. So here, after completing a course, you will achieve 500 points. After running a pipeline, you will gain 100 points, and updating your profile will give you 100 points. And finally, group activities can earn you 10 points. After achieving the learning outcomes, you would have learned some of the major best practices in the field of bioinformatics. You will be exposed to R and Python, two powerful programming languages out there. Bioinformaticians will need to understand how to use t-test, regression, and other tests. Analyzing and managing data in order to communicate important information is another best practice in bioinformatics. Pipeline development and utilization is another common practice. Ultimately, completing the precision medicine program will help you enter the field of bioinformatics since you learn several best practices. Now, I would like to take you all through the sign up Omics Logic Learn portal. So here, since I have already signed up on the portal, I have my email ID and password saved on the portal. Otherwise, you can click on create an account and enter your name, email ID, and choose a password for the account. Or you also have the option to sign up using any of the social media handles, that is Google, Facebook, GitHub, or Twitter. Since I'm already signed in using the my Gmail account, I'm going to proceed further. as explained, click on the tab that displays your email ID. This will take you to your profile. So here you need to have an updated profile picture. Your name should be capitalized in proper manner. You have to link your social media handles, include your educational background and your research interests. All right, so the next, I would like to invite Mr. Ilya Brodsky. His background is in applied mathematics and data science. He has facilitated and managed collaboration with researchers at Stanford, UCSF, Louisiana State University, and other academic and pharma research and development teams. Mr. Brodsky oversees the strategic development of research projects and manages the team responsible for bioinformatic analysis, training, and collaborations for Fine Biotech. In this part of the webinar, Mr. Ilya Brodsky will be discussing about the overview of the program. So over to you, Mr. Ilya Brodsky.
Thank you very much, Sri Gauri, and welcome everyone to this webinar. In my part of this presentation, I want to briefly introduce the topics that we will cover in the training program to give you a sense of what kind of activities you will be involved in, what kind of topics you might apply bioinformatics to, and how will we structure this program. And then I'll pass it on to another team member to talk about the structure of each session, what is going to be covered when, how to register for the specific program, and what to expect. So when we speak about bioinformatics, typically the background of the majority of the people related to bioinformatics is in research. And in research, primarily the focus of bioinformatics is to understand what is in the data and link it to a specific phenotype so that we can characterize a very defined process and find links to the application. And then bioinformatics in medicine has to do with taking that research and applying it to diagnose and treat specific diseases. And in this program, we're going to focus on several different diseases. One of them, and a major cause of death in the United States and around the world, is cancer. So cancer was the second leading cause of death in the United States, or in the world, sorry. And primarily it affects Western nations, but it is affecting more and more developing nations as well. And the study of cancer has led us to, first of all, kind of identify specific genes that are responsible for different aspects of cancer development and progress. And then eventually today, more and more studies focus on a much greater detail of how the systems biology of cancer can be used to treat and help people even by preventing cancer. So to understand cancer progression, we need to understand that healthy cells, as they develop, eventually some of them lose some mechanisms of restriction that cause those cancers with damaged DNA and all kinds of unwanted processes to proliferate uncontrollably and even spread throughout the body. And so when we speak about cancer and stages of cancer development, we're speaking about the onset of cancer, when these cells start to appear and there's a tumor, and eventually spreading throughout the body, and that is what we call metastases. And what's important to address this problem, to really treat cancer, a critical part of that is screening and identifying the risky populations, and then within those risky populations to identify the onset of cancer. And so what people have been doing to understand these mechanisms at the biological level is identifying markers of cancer risk and translating those markers of cancer risk to strategies for screening and early diagnosis of cancer. Now, a lot of that relies on modern technologies like next generation sequencing. And what people have found is that next generation sequencing can allow us to understand what happens in the cell using the same technology, but monitoring several different critical molecular processes inside tissues and cells. And collectively, we call these data types generated by next generation sequencing omics. And these omics data can be applied to different levels of what happens within the cell on aggregate, for example, in a whole tissue, or specific to an individual cell. And that's where we have single cell technologies like single cell genomics, single cell transcriptomics, etc. And so when we try to understand how these technologies can help us detect what's happening inside the cell, one of the ways to use is to measure transcriptomic signals. And those signals are a good proxy for what we will call as the functional component, the proteins that are being produced within the cell. Here's a good example. In breast cancer, there is a specific receptor on the surface of the cell called HER2. And that receptor is responsible for signaling that communicates with inside the nucleus how to produce specific growth hormones. And that is one of the causes of this unproliferated growth. Well, to measure proteins on the surface is not very practical. And so people have used transcriptomics to identify the gene that expresses HER2. And that gene is ERBB2. And by looking both at the level of expression and alternative splicing of that gene, we can identify potential responders to treatment. But these kinds of strategies focused on just a single gene have essentially been evolving. And so what we see within different types of cancer, like for example, breast cancer, we are seeing that the classification of cancer continues to expand. And it continues to expand based on a growing number of marker genes like HER2 and an integration between those genes, which is the molecular classification, but also multiple other factors like even proteomics and metabolomics. And that's why we see this conversion between research-based bioinformatics and clinical use of bioinformatics, where this kind of understanding of what happens at the molecular level is actually critical for accurate classification of an individual patient or a group of patients, but ultimately also for selecting a treatment that is more likely to work and be effective for that individual. So as a result, bioinformatics for clinical use relies on big data of various types that is integrated and ultimately is being used for diagnostics and therapeutics. Now, what's interesting is that not only is this data being used for patients, but essentially we are seeing a number of experiments that are not possible on actual human patients. And so another major challenge in these kinds of studies is to be able to accurately represent what happens inside the patient's body by using animal models of human disease. And so when we will explore the role of bioinformatics in precision medicine and examples of bioinformatics applied to data sets, we will use both clinical samples taken from patients, but also animal models like patient xenograft models, where a tumor is grafted inside the animal to study those biological processes, but also to try and intervene using a variety of compounds. And that essentially leads us to this kind of big data paradigm in molecular studies, where we have kind of an approach of using data to inform our hypotheses and essentially then being able to model a specific process and apply that process to actual treatment of patients. And that leads to shorter discovery and validation timeline, higher success rate, and lower costs. And these are really the objectives of precision medicine in terms of their actual clinical use. Because if we were just to take all of the data available about the patient and use that data for every single individual, it is just simply cost prohibitive. But if this kind of collection and analysis of data is informed by large scale studies, that then can stratify patients and determines which ones are likely to benefit from this approach, then it leads to actual improvement in outcomes, more efficacy, but also lowering costs overall. So in this program, we will benefit from those large studies that have collected systematically data about different types of projects. And so as you can see on this chart, the growth in the amount and availability of data has been exponential over the past couple of decades, actually. And today we have access to smaller studies and larger studies. And one of those studies related to cancer is called the Cancer Genome Atlas, Tcga. So what's unique about this study is that they have been able to collect systematically from different locations and in different types of tissues and cancers, a huge collection of thousands of individuals that comprehensively characterizes their genomic, transcriptomic, proteomic, and even metabolomic profiles, allowing us to search through the clinical data that are associated with risk factors and outcomes and even treatment, and determine how we can use this data both at the clinical and the molecular level to better understand this complex disease. So one of the studies that we will look at is how such multi-omics integration and analysis of molecular profiles has led to a new understanding of complex diseases based on multiple types of omics data. But the TCGA project is already not so new. So I think it was started in 2013, so it's been around for quite some time. What has happened since then? Well, more recently, a new study called All of Us Research has started making their data accessible as well. And so there we have, the objective is to have over 1 million individuals that represent both different kinds of conditions, but also help us understand the variability of healthy individuals. So another source of data that we will look at, and we will try to see what kinds of cohorts are available, is the All of Us Research Hub. And I think for all of you that are coming from a research background, this is an exciting new opportunity to try and understand and develop preliminary hypotheses based on this data about those topics that you are interested in. Another interesting project that we will look at throughout this program is the NIH Human Microbiome Project that exclusively focuses on characterization of metagenomic sequencing for human microbiome, both for healthy, clinical, and preclinical individuals. It's an exciting project that has grown over the years. Today we have the Human Microbiome Project 1, Human Microbiome Project 2, and the project continues to evolve with specific focuses and actually is going to be a part of the All of Us research as well. In addition to these large repositories, we will also look at several examples of publications that have leveraged animal studies or cell line studies to accurately represent patients and test different assumptions, both about molecular characterization of patients, but also on testing and response to different types of treatments, both those that are already on the market and those that are still being evaluated in clinical trials. Now, when you think about all of these different data sets and the skills that you might need to have to address some of the interesting questions that you could answer with these data sets, you might feel overwhelmed. And this is understandable because there are so many different tools and packages and methods, including statistical analysis, machine learning, and deep learning. It is difficult to envision one to be able to go through all of that, learning from scratch about the important questions, the data sets, and all of the tools. And so for that reason, what our team has done is worked with the Tcga Bioinformatics Research Center that has developed a user-friendly version of all of these complex algorithms that one can learn to use and understand the logic of analysis without being hindered by the technical complexity of each one of these methods. So the platform offers a user-friendly interface. On this interface, there are color-coded paths or pipelines that you can reproduce accurately and get easy to understand and use visual outputs that speak to the biology that can be found in this data. Included are several different pipelines and utilities that you can use to work with this complex data, be able to understand and mine it, and especially we will use curated data sets that allow us to deploy standard or conventional machine learning techniques to explore and understand variability, defining groups of patients or specific gene sets that can identify individual patients. Now, what we'll do is we will look at this through the lens of case studies. And we will try to use data from the types of studies that I already mentioned, like cancer, human microbiome project, or All of Us research, but we will also incorporate some ideas from the infectious diseases, where an important trend has emerged over the past several pandemics or epidemics in the past couple of decades. And those include such outbreaks like Ebola virus outbreak in West Africa in 2014, 2016, different types of understandings of the disease, the pathogen interacting with the host and understanding responses to those markers, but also using microbiome and infection, which will allow us to appreciate the role of multi-omics integration and bioinformatics in those kinds of studies as well. So what can you expect as a result of going through such training? The real outcome is a project. Not only will you be able to listen to lectures from experts that will highlight some important aspects of current and traditional development of bioinformatics in different applications, but you will also be able to design a project that highlights and showcases to the world what you are able to do with these kinds of skills. So specifically, we will talk about the application of such tools for clinical decision-making, where we will look at such applications like patient stratification, disease classification, identification of potential responders to therapies, mechanisms of disease onset and progression, and the way that such tools are used in both pharma and biotech early biomarker discovery, detection of toxicity at the in vitro stage, analysis of molecular mechanisms of a drug action, and drug repurposing or repositioning. So these kinds of topics are impactful and can be very meaningful, whether you are a student and you would like to demonstrate what you have learned and how you have been able to use data to ask and answer some of these complex questions, or they could be useful for your research, where you're interested to get into this field, you would like to improve your skills, but ultimately you would like to try and see how they work in real life. Throughout the program, you will be given access to some of the tools that I already mentioned, but we will also rely on RStudio and BioPython to analyze data sets and write the code, maybe simply by modifying it or actually writing it from scratch. And to do that, our portal that Tcga already showed to you and you have enrolled in offers a number of guided processes to help you understand the link behind the logic of the analysis, the syntax of the code, the different curated data set examples that we are going to look at, and practicing that code so that you can ultimately become independent. And as a result of this, you will be able to accumulate a repository of your own code that you can essentially use in the future for your own research or for whatever situation you will come across in your future career. So here are some learning outcomes that we envision for this program. I think that you will learn to appreciate and understand scientific literature that describes analysis of various types of omics data, including genomics, transcriptomics, metagenomics, and others. You will know how to perform basic processing steps for such data with consistent accuracy. You'll be able to perform exploratory analysis and generate a hypothesis from data. You'll know how to design contrasts for statistical tests and prepare data for machine learning. You'll be able to annotate found signal using biological databases and references. And you learn how to identify appropriate data sets for answering a specific biological question. As a result, you will learn and be able to plan a bioinformatics analysis from start to finish completely independently. And we will offer an opportunity to present your final project proposal, including the rationale, the methods, scientific background, and anticipated results to an experienced mentor that will be able to provide you with feedback. I'll finish with this testimonial of a previous participant. Her name is Olga Udartseva, and she was the research affiliate, a postdoctoral student at Roswell Park Comprehensive Cancer Center. What she said was that the program is very well structured and easy to follow. The skills that she gained through the program helped her significantly enhance the relevance of her research by making connections between the preclinical study and its practical application to cancer patients. So with that, I would like to pass this on to Dr. Mohit Mazumdar, who will speak about some of the program resources, the outline of the different sessions, and all of the topics that will be covered in those sessions. Over to you, Mohit.
Thank you, Ilya, and hello, everyone. I would like to share my screen first. And I would like to say hello to all of you here who have joined this webinar. And today we would want to, as Sri Gauri introduced us and told us about the flow of the webinar, we wanted to show you an overview. Followed by that, the next segment is to tell you more about the structure of the program and how the sessions will be conducted and to answer any questions that you have for this training. So I'll start with my introduction. So my name is Mohit, and I have a PhD in computational biology and over 40 publications on various topics, research problems that I was able to collaborate and work with different scientific groups. And in Pine Biotech, we are helping students to achieve some of those outcomes through specific sort of training, which are very targeted. And today we are going to discuss this great opportunity to learn about a very innovative, incoming, and very important field of life science, which is precision medicine. And it's training that we have designed that is something that you will not be able to find elsewhere. So about the sessions, the way we have scheduled this entire program includes a lot of resources that you can learn and take on your own. So you will have your own time at which you can learn from these resources, which we will be sharing throughout the program. However, we will also have live sessions, which are like 10 live sessions, which we have scheduled for the month of June and July, where we will be discussing about these several topics, and you'll have an opportunity to discuss and ask the mentors while we go through these topics and discuss several of these projects and case studies that Ilya was mentioning. But before that, I think the best place for me to show you this is the Learn page, where we have put all of this together. So if anyone can confirm that, if Sonalika, if you can confirm, or Ilya, you can confirm if you can see my browser as well.
Yeah, we can see it. All
right, perfect. So
yes,
as I was telling you that this is the first orientation session where we wanted to take you through the program and tell you that why we are conducting this program, what are the benefits, and what we are going to go through this in this entire research-based experience. So it's very important to understand that what will be the flow, when will be the sessions held, and how much access you will have for all of these resources. So we have put together this program page on Learn Omics Logic, and this is the link for this page that you'll find in the chat. So in here, we have described the sessions with dates and the topics that will be covered. Along with that, very interestingly, you'll find the associated resources hyperlinked in this page. And this is a great place to overview the entire program, because we will have the first session on 14th of June, which will be the start of the program, where we will be discussing and taking you through the specific specialized sessions with expert. But before that, the time that you have, we will encourage you to take the program and start with some of the preparatory materials that we'll be sharing once you register. So we will talk about the registration towards the end of this webinar, where we will be taking you through the different subscription levels that we have and the benefits within those levels. But before that, let's discuss the sessions one by one and the resources that it associates with it. So the first session, we are going to start with a major problem, the cancer research problem and cancer problem that Ilya was describing in much detail. So without taking much of your time, I'll just like to tell you that we are going to start with some of these case studies where a lot of NGS, a lot of data science, and a lot of other technologies are used. So we have these different examples for you to learn from. So we will be discussing about the complexity of cancer and where bioinformatics is used, where this entire big data is being how this data is being generated, and then what are the different ways we can visualize this data, understand what is in there, and where to look for. So we have also included some of these project examples for you to get started and understand and look at the overview. So if you click on some of these links and open it in a new tab, so you'll find out that what kind of material is associated with one of these lessons. So I already have a subscription here, so I've been able to go into each of these courses and see that how a detailed lesson is provided within these links. Now, after the first session, where we'll be introducing about the cancer problem, and where the application is, we will learn about some of the emerging and important technologies through which we can find some of the interesting facts that Ilya was describing about finding specific mutations, understanding how to analyze genomic data. So the next session, which would be on 16th of June, we will be discussing about the next generation sequencing method and application of genomics, where we will be learning about, from the very beginner level, we have materials put together on genomics. So there is a course 3 on Learn Omics Logic that describes the genomics from a DNA point, from a beginner level, where you're learning about DNA and its structure, to the big data and how do you interpret, how do you map variants, how do you detect and find those small changes, which is equivalent to finding needle in a haystack. So the first session, after the first session and the second session, which would be on genomics, we will be going through a case study. And this is a very interesting case study on oncogene variant calling. And again, you can always go to this page and check for yourself these links, and go to these links and understand that what this project would be all about and what this session will be about and what we'll be covering. So here there's an example of a new project on oncogene variant calling that we'll be covering in this case study session, where we will be discussing about understanding that how mutations and variations accumulate with time and are passed on to the progeny, and then how do you find this prominent mutation that could help you find answers to some of the major problems when we are looking at a strategic plan to stop or design clinical trials for these cancer studies. So understanding that would be understanding genomics and understanding how genomics can be applied and how genomics is applied in real research. So it's a part of omics logic training that you have to go through some of those data sets that we have put together from high value publications, from peer-reviewed articles, and also data sets that are really digested well in the community and have a translational research outcome. Followed by that, we have a session on 23rd, which describes the transcriptomics data analysis. Because when we are talking about precision medicine, we are describing that how different types of data helps in understanding how we can come up with a solution strategy which is much personalized and helps in finding those answers within small cohorts. So here we are looking at gene expression data. We are trying to understand that how do you interpret and analyze gene expression data using statistics, using machine learning, unsupervised machine learning. So we will start understanding about analyzing the transcriptomics data using different algorithms and workflows on Bioinfo platform, as well as applying downstream analysis processes to visualize, to understand the nature of the data and the patterns from the data using machine learning methods. So from here on, we will be learning about different machine learning methods that could be utilized in understanding and visualizing and interpreting the data that is generated from these experiments, where we have patient samples, where we have cell line data, where we have data from mouse models, and all of these data resources is something that are playing huge role in everyday's research, in like cutting edge research. So how do you find these data sets is something that we are going to cover in the next session, which would be an amazing session, because understanding how to analyze data and then finding data kind of summarizes it all, that how one can go about a new research, how one can develop a research question. So these 5 sessions are going to cover some of the major aspects, including some of these important data repositories such as NCBI SRA, NCBI GEO, and Tcga Cosmic, and all of those that you might also be interested of. And you can also be a part of these sessions by registering for the program and ask and add your own areas and discuss with us, and so that we can help you in understanding more. The next session is again a case study on modeling cancer precision medicine, where we will be taking you through this project. You can obviously take a look at this link also. So this project tells about breast cancer data again, and finding genomic mutations, and understanding that how different types of responses to treatments are integrated with different types of omics data. So this will include machine learning as well as use of omics data and integrating the drug response, so that you understand that how this is applied in precision medicine, which is very relevant to the program. So after this session, we have a session which is more focused towards clinical diagnosis, because we are talking about precision medicine. So we are talking about different types of challenges that the world is facing, which includes multi-drug resistance, which includes finding stage specific interventions and all of that. So we'll be learning about different case studies here. We will be learning about how viruses and how bacteria, how pathogens are also causing some of the major diseases, which we were not able to correlate before without the analysis of big data. But with the help of big data and different types of big data, we've been able to analyze and correlate and understand that how this plays a role in clinical diagnosis. Now, as we were discussing before, and we went through this entire overview by Ilya Brodsky, where we learned about that not only cancer, but a lot of analysis, and especially after the COVID outbreak, has been done for infectious disease research, where precision medicine is playing a vital role. So from cancer, we will move to another important aspect of human health, which is infectious diseases. Here we will learn about host response to viral infection. Then we will learn about the COVID-19 case study that we have put together. So there is also a project that we will be discussing in this session, and you can also check this project 2 that we have on COVID, which is available on Learn portal, where we will be discussing about as we discussed also, like how this affects the host, and also what are the different transcriptional signature when we have a treatment. So understanding from a drug response level to understanding how at a genomic level this plays a role when we are looking at the genome of the virus itself. So, very exciting sessions that we have put together. So after July 14th, we have July 19th, which is like the final session, where we are going to discuss how all of this information makes sense for you to work on your own project. Different case studies, putting together a logic from developing from introduction to developing a research question and finding results, and then validating it and trying to work on a research project. So advancements in precision medicines, areas and opportunities, and also like this includes the opportunities that are there with which we are sharing regularly on our social media handles. So this is what we have put together for you to learn in this summer program, which is Bioinformatics for Precision Medicine. And I'll be happy to answer any of questions so far if you have about the schedule, about the sessions, about the timing, about the time commitment that you would need to go through all of this. So please feel free to ask your questions in the chat, and I'll be happy to answer those. Next, we will be taking you through the registration process and telling you what different subscription models that we have that actually helps every individual participant to get this personalized experience for learning precision medicine and application of bioinformatics in precision medicine.
Thank you so much, Dr. Mohit. And we have a question by Abdul. Are these all lectures, or will we be? Yes, they will definitely include hands-on practical sessions as well, along with the lectures, Abdul. So the program page that Dr. Mohit just went through and explained each and every session's topics, in the program page itself, we have listed the practical sessions as well that you're supposed to go along with the lecture series. All right, so I'll just take you through the program resources, and then we'll discuss about the registration process.
Hold on. There are a couple more questions. Sonika, a couple more questions. So Gully VT is asking, is it suitable for computer science research scholar? And Pragna, how much time do we need to dedicate per week? So Mohit, maybe you can answer those questions.
Yes, sure. So for a computer science research scholar, I think we have already students who have come from computer science background and are part of the program. So for those who are from computer science background, we do understand that they need a overview of biology and overview of application of these biological data analysis. So we have prepared the asynchronous coursework, Galivetti, which helps you, to understand some of those basic concepts so that you can learn and apply meaningful and do meaningful research in this space. So I think, yes, if you are thinking about doing some of these omics logic training programs, I think we will be able to help you go through the training. The second question is, I see, is from Pragna. How much time do we need to dedicate per week? So per week, we have 2 sessions, and the sessions are usually for 60 minutes to 90 minutes, which includes like overview of the concept that we are covering. And then there are practical hands-on session, and then there is a Q&A session. So you will be having this opportunity to discuss with the mentor also what is being covered and if there are any questions. But in addition to that, as I was showing you that asynchronous resources and materials that we have, so during the week, you will have to dedicate at least 3 to 4 hours in the beginning when we are starting the program to go through some of those resources, and then you can reduce this time based on your availability and having the sessions and all of that. But overall, I would say you'll have to take out 5 to 6 hours or 7 to 8 hours based on your expertise and level for this training program per week. Okay, there is another question by Spandan. So is this program manageable with the 3-month program that we might have enrolled for already? So if you have already enrolled for a 3-month program, I'm not sure which program you're mentioning, but I think we have participants who are taking multiple programs, and it is actually helping them to find some of the problems. If you're working as a research fellow and if you want to do a specific training on precision medicine, and if your research proposal is on precision medicine, I think it will help you to take the program, take this program and directly work on these case studies and problems which are very specific to the topic that we are covering. So yes, I think this will be helpful and manageable if you have time during the summer, and if you can attend the live sessions, complete the assignments, I think it's manageable.
All right, so I think that you have answered all the questions, Dr. Mohit. Thank you for that. And now I would like to take all the participants here through the program resources and also the steps to register themselves on the program page. So first of all, I'd just like to introduce myself. I'm Sonalika Ray, and I'm the Omics Logic Community Manager, and I'm responsible for the daily interaction with thousands of our users that are a part of a community that is managed by Pine Biotech. So for the different parts of the sessions that Dr. Mohit explained, we will leverage Omics Logic resources that you can use to learn about precision medicine. We'll explore the asynchronous coursework with theoretical material, quizzes, and practical assignments. Our cloud-based analytical server where you can analyze large omics data and visualize results. The Omics Logic Code Playground will help you practice coding in R and Python, and as well as repositories of project examples are some of the things that we're going to explore. These methods, which we discussed, will include hands-on exercises on the Bioinfo platform, which provides a simple and intuitive user interface for processing, analyzing, and machine learning methods for multi-omics data. And throughout the course, you will be given the directions to access the Bioinfo platform for bioinformatics processing and analysis of RNA-seq data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of analysis are performed in different sections in this multi-omics platform. And behind many of the methods are scripts written in popular coding languages such as R and Python. And these are also explained in tutorials that you will access to from by being a part of this precision medicine program. So from visualization of genomic data to exploratory analysis and hypothesis testing. At the end of the program, participants will receive the certification of participation and also registering for the program. Now you will get the access to the limited scholarship seats that we have that would enable up to 50% off on the program registration. I will shortly take you through the program page and explain the steps to register yourself for the program. Also, I will take you through our social media handles, which you can join and become a part of our bioinformatics community. In these social media handles, we keep sharing about recent trends in research, training material to learn and get hands-on practical skills for these trends, updates about Omics Logic Learn portal and also the Bioinfo server. Also, we share job posts for those who complete the Omics Logic training and are ready to apply their knowledge to resolve the real-world problems. So I'll just quickly go to the Omics Logic Learn portal now and take you all through the program page and let you know how you can register yourself for the program. So once again, we are back on the Omics Logic Learn portal. The link is in the chat. Yes. Now, please make sure that you have signed up already and have an account, as Shri already explained, and have updated your profile. Now, let's go directly to the program page and see how you can register yourself for the program. So on the program page, you will find all the information. If you have any questions, if you still have any questions about any of the topics that would be covered, you can go through the syllabus here on the program page. And if you scroll a little bit more, you will find 3 different levels, which are beginner, intermediate, and advanced. These levels actually refer to the number of days that the participant will have an access to. So in the beginner level, you're going to have the access to the program for 45 days. And in the intermediate level, you have an access to the program for 16 days, where you will also be able to come up with a research proposal that you can further work on in the research fellowship program. And in the advanced level, you will get the access for 90 days, where you can work on your research project as well, along with the precision medicine training. So that is the difference between beginner, intermediate, and advanced level. And also in the intermediate and advanced level, you will get the access to one-on-one expert sessions and 24/7 mentor support. In the advanced level, you also have an option for certificate of excellence, as you'll also be a part of the research fellowship program. Now it's up to you for how many days you want to enroll for the program. And it's very simple to register yourself. You just need to click on the select option button for whichever subscription level you would like to enroll yourself for, and then just proceed with the registration. Also, here on the program page itself, we have listed some program courses and project libraries that you will have an access to as a part of this program. And also you're supposed to go through these resources. So as Dr. Mohit explained that we have a bunch of example projects that you will be going through and learn from them about how precision medicine could actually be applied. So these are the resources that you can go through and learn from. And after a successful completion of the program, you will be getting a certificate of completion and also a certificate of excellence as a part of the advanced level of subscription that I already explained. Now I want to share with you some of our social media handles so that you can stay updated with all the updates and all the training programs that we would be having, and also be updated about job posts that we keep on posting on our social media handles. So our presence is there on YouTube. I'll share the link in the chat as well. And do join our community on YouTube so that you stay connected with us and watch the videos that we keep posting about different types of specialization tracks and also the training programs. And then we are also there on Facebook by the name of Biotech. So you can follow us on Facebook as well if you are there. And now comes the very important groups on LinkedIn. So I'll just share the group of our LinkedIn channel as well in the chat box. So we have a page on LinkedIn by the name of Omics Logic, and we also have a group where we have experts as well, and we keep on posting about the updates and also the resources that you can leverage from. This is the page that is Omics Logic, for which I have shared the link in the chat. And the group's name on LinkedIn for Omics Logic is Machine Learning, Biomedical Data Mining, and High-Throughput Data Analysis. So you're most welcome to join our bioinformatics community and leverage the resources and also the experts who are a part of a community. So that was all that I wanted to discuss with all of you today. And I see we have a question that Sparsh has already answered. That's great. So do we have any other questions? Please, you can put them in the chat box.
So Nalika, I don't see any more questions, and I'd like to thank everyone for joining today's session. We will also be sharing the session video recording.
All right. So if we, yes, sir. So if we have no more questions, I think we can end the session. And before we end today's session, I just like to put our email address, that is marketing@omicslogic.com. Please feel free to reach out to us for any questions, and we'll be happy to help. And while enrolling, if you face any technical issue, please feel free to reach out to Sparsh. She will help you become a part of the program and get started with your training and research in precision medicine. Next, we'll be meeting on June 14th. So thank you everyone for joining. We'll catch up on June 14th when the program begins.